Proteins are the molecular machines of life, folding into precise three-dimensional shapes to perform their biological functions. But what happens when this intricate folding process goes wrong? Why does misfolding sometimes cause devastating diseases, while in other cases it triggers protective mechanisms? This paradox challenges our understanding of protein biology and cellular homeostasis. One of the most well-known consequences of protein misfolding is the formation of harmful aggregates, which can lead to neurodegenerative diseases like Alzheimer's, Parkinson's, and Huntington's disease. In these conditions, misfolded proteins clump together into amyloid fibrils, which can disrupt cell function and ultimately cause cell death. Why doesn't the cell just destroy these misfolded proteins? Normally, cells rely on quality control systems such as chaperones and proteasomes to refold or degrade damaged proteins. But when the system gets overwhelmed, toxic aggregates accumulate, leading to disease. Yet not all misfolded proteins are bad. In some cases, misfolding can activate protective cellular responses. For example, cells can initiate the unfolded protein response, UPR, which enhances the production of chaperones to restore proper folding. In stressful environments, certain misfolded proteins even act as signals to help cells adapt to changing conditions. Could misfolding, in some cases, be an evolutionary advantage? Remarkably, some bacteria and yeast intentionally use misfolded proteins as part of their survival strategy, storing proteins in a partially unfolded state that can be rapidly refolded when needed. A fascinating example of beneficial misfolding occurs in prions, a unique class of proteins that can exist in both functional and harmful forms. While prion diseases such as mad cow disease result from toxic misfolded prions, some organisms use prions for adaptive advantages. In yeast, prion-like proteins help cells survive environmental stress by triggering beneficial changes in gene expression. Misfolding can also influence immune system function. In some cases, protein aggregates help the immune system recognize pathogens more effectively, acting as natural adjuvants. This paradox raises an intriguing question. Could certain diseases associated with protein misfolding, like Alzheimer's, have originally evolved as unintended side effects of protective responses? The protein misfolding paradox forces us to rethink our approach to treating misfolding diseases. Instead of always trying to eliminate misfolded proteins, could there be ways to harness their protective functions? As researchers explore new therapies, including drugs that modulate protein folding pathways, could we learn to control misfolding rather than just fight it? Understanding this paradox could pave the way for breakthroughs in neurodegenerative disease treatment, aging research, and synthetic biology, offering new strategies for manipulating protein behavior in both health and disease. Proteins must fold into specific three-dimensional structures to function correctly, but errors in folding can lead to misfolded proteins that aggregate into harmful structures such as amyloid fibrils, which are associated with neurodegenerative diseases like Alzheimer's and Parkinson's. However, some misfolded proteins trigger protective cellular mechanisms that help manage stress and restore homeostasis. In Huntington's disease, misfolded Huntington protein forms toxic aggregates, whereas in yeast, prion-like misfolded proteins can provide an adaptive advantage under stress. This is like a machine assembling a complex puzzle. If one piece is placed incorrectly, it can jam the mechanism, but in some cases, alternative configurations create a new, functional system. Cells use molecular chaperones, such as heat shock proteins, HSPs, to assist in proper protein folding and prevent aggregation. These chaperones recognize misfolded proteins and either refold them or mark them for degradation. In some cases, misfolded proteins stimulate the increased production of chaperones, enhancing the cell's ability to manage future stress. HSP70 prevents the aggregation of as-synuclein in Parkinson's disease, but its overexpression in some systems enhances stress resilience. Chaperones act like quality control inspectors in a factory, ensuring defective products are either repaired or discarded before they cause harm. While many protein aggregates are toxic, some organisms naturally form functional amyloids that serve beneficial biological roles. These functional amyloids are structured, stable, and used in processes like bacterial biofilm formation and hormone storage. The distinction between harmful and functional aggregates depends on structural organization, cellular environment, and regulatory mechanisms. Mammalian hormone storage relies on amyloid-like structures, while amyloid DA aggregates in Alzheimer's disease lead to neuronal toxicity. This is like bricks in construction. When arranged correctly, they form a sturdy building, but when haphazardly piled, they create an unstable structure. Some misfolded proteins act as cellular stress sensors that initiate protective pathways, such as the unfolded protein response, UpR. These responses upregulate stress-handling proteins, enhance proteostasis networks, and help maintain cellular function under challenging conditions. However, prolonged activation of these pathways can lead to cell death if misfolding persists. The endoplasmic reticulum stress response triggered by misfolded proteins can either protect cells from damage or contribute to diseases like diabetes and cancer. This is like a fire alarm. When functioning properly, it warns of danger and prompts protective actions, but if it keeps triggering, it can lead to overreaction and system failure. Prions are self-propagating misfolded proteins that can cause neurodegenerative diseases, but some prion-like proteins serve beneficial roles in normal cellular function. In yeast and bacteria, prion-like proteins regulate gene expression and cellular adaptation, showing that misfolded states can be evolutionarily advantageous. This suggests that prion mechanisms may have initially evolved for beneficial purposes before becoming pathological in mammals. The PSI plus prion in yeast allows for rapid phenotypic variation under stress, enhancing survival. Prions are like viral memes. Once introduced, they spread and influence behavior, sometimes destructively, but sometimes leading to innovation. Misfolded proteins can act as damage-associated molecular patterns that stimulate immune responses. While this can lead to chronic inflammation in diseases like Alzheimer's, controlled immune activation helps clear cellular debris and protect tissues. This dual role highlights the fine balance between protective and pathological responses to misfolded proteins. Amyloid beta aggregates trigger neuroinflammation, which initially helps clear debris but eventually contributes to neuronal damage. This is like a city's waste management system. If it works efficiently, it keeps the streets clean, but if overwhelmed, it leads to pollution and chaos. Certain organisms use controlled protein misfolding as an evolutionary strategy to generate diversity. In stressful environments, increased protein misfolding rates can introduce new functional properties which natural selection can act upon. This suggests that, in some contexts, protein misfolding may drive adaptive evolution rather than purely being a pathological event. In bacteria, stress-induced protein misfolding alters enzyme activity, leading to novel metabolic capabilities. This is like genetic mutations. Most are harmful, but occasionally they lead to beneficial adaptations that enhance survival. As organisms age, cellular proteostasis networks decline, leading to increased protein misfolding and aggregation. Aging diseases such as Alzheimer's and ALS arise when the balance between protein synthesis, folding, and degradation is disrupted. However, some long-lived organisms exhibit enhanced proteostasis mechanisms that mitigate misfolding effects, suggesting potential anti-aging strategies. Naked mole rats show increased autophagy and chaperone activity, reducing protein misfolding despite long lifespans. This is like an old library. Without proper upkeep, books become damaged, but some libraries implement conservation strategies to preserve their collections. Understanding the paradoxical roles of protein misfolding opens avenues for developing therapeutics that modulate misfolding pathways. Small molecules, gene therapies, and chaperone-based treatments are being explored to either prevent harmful aggregation or harness beneficial misfolding. This research holds promise for neurodegenerative diseases, cancer, and synthetic biology applications. Molecular chaperone activators are being tested in clinical trials to slow Parkinson's disease progression. This is like engineering traffic control, redirecting congested pathways to optimize flow and prevent blockages.